Okay, good afternoon, everybody. Good afternoon. So, welcome to today's class. My name is Madam Lillian Cherono, and I'll take you through microbiology techniques. Can you hear me? So, our session today is staining. So, I want us to learn about staining. So, who can define for us the term staining? What is staining?
Okay, so let me try. Okay, so staining is a method used to ensure that the microorganisms being observed are visible in the microscope.
Yeah, staining is the process. Staining is the process of coloring the structure of cells, tissues of microorganisms to enhance visibility and contrast under the microscope. So the microorganisms cannot be visible if not stained when viewed under the microscope. So in microbiology, we have two types of staining. We have simple staining and we have indirect staining. Simple staining involves the use of a single dye to color the cell structure tissues of microorganisms. For example, the use of iodine solution or methylene blue to color the thin epidermal layer of the onion bulb so as to study the structure of the plant cells. So in simple staining, we use a single dye and therefore it can also be referred to as direct staining. Then we have the indirect staining, which involves the use of several dyes, and a good example is gram staining. So gram staining is a differential stain which differentiates the two groups of bacteria. So can someone give me the two groups of bacteria based on the cell wall classification? Someone to give me the two groups of bacteria based on cell wall classification?
We have gram positive and gram negative.
Okay, very good. So we have gram positive bacteria and gram negative bacteria. Who can give us the difference between the two types? Joan?
Yes, madam. Gram positive bacteria have a thick cell wall, while gram negative have a thin cell wall.
Okay, that is good. So gram positive bacteria, they have a thick cell wall, while gram negative bacteria, they have a thick cell wall. So we said bacterial cell wall is made of a compound known as peptidoglycan. So the gram positive bacteria, they have that thick layer of gram positive bacteria. They have thick layer of peptidoglycan layer, while gram negative, they have a thin layer of peptidoglycan layer. So in addition to that particular compound in gram negative, they also have lipopolysaccharides and phospholipids. So in gram staining, we use different reagents or chemicals. So who can give me the examples of the reagents we use in gram staining?
We have crystal violet.
Yeah, that's good. We have crystal violet. Joan? Joan? Joan, I can't hear you. Joan, can you unmute and speak? So I can't hear Joan. Francisca?
Safrani. Okay, we have safrani.
Yeah, we have safranin. Jacqueline? So I can't hear you, Jacqueline.
Methylene blue.
Okay, so we don't have methylene blue. So the list of the reagents which we use in gram staining, we have primary stain, which is the crystal violet, and it is purple in color. Then we have the mordant, that is the grams iodine. And then we have the decolorizer, which can be acetone or alcohol. And lastly, we have the counter stain, which can be safranin or neutral red. Okay, when you observe under the microscope a specimen of microorganism, when you suspect to be gram positive, which color are you supposed to see under the microscope? Joan or Winnie? Which color are you supposed to see under the microscope?
It retains the purple color of.
Yeah, that's good. So if the bacteria is suspected to be gram positive, it will retain the purple color of the primary stain, that is the crystal violet. What of gram negative? What of the gram negative, Otieno?
I think it doesn't retain the color.
Yeah, it will retain the color, but instead it will take the color of the counter stain. That is the color of safranin or neutral red. Therefore, it will appear red or pink when you observe under the microscope. So we have learned the reagents which are used during the gram staining. So for your assignment, I want to give you the assignment. You go and look at the principles of the gram staining and the procedure for carrying out the gram staining. So I will look at the assignment in our next class before we begin the session so that it can enable us to carry out a practical concerning the gram staining. So make sure you do the assignment. You look at the roles of the reagents, the principles, and the procedure of the gram staining. Otherwise, is there any question?
No,
Anyone?
we do have a
So,
question.
so if
What are
there's
the
no,
examples of gram positive?
okay, the examples of gram-positive bacteria, we have the Staphylococcus species. You can say Staphylococcus aureus. We have Streptococcus species, Lactobacillus, among the others. Examples of gram-negative bacteria, we can have Salmonella, each is Salmonella typhi, Vibrio cholerae. We can have E. coli. Also, we can have Clostridium, among the others. But still, you can add on the list of both gram-positive and gram-negative. Sawa sawa. Otherwise, thank you for joining the class and have a lovely afternoon.
Thank you, madam.
Thank you.
Bye.
Thank you.
Bye.
Bye.